Hello friends. In today's video, we'll continue with Food Microbiology, Module 1, the Historical Development of Microbiology and the Evolution of Microscopy, Part 2. We'll explore the groundbreaking contributions of world-renowned microbiologists who shaped this field. Antony Van Leeuwenhoek, a Dutch tradesman and self-taught scientist, is universally recognized as the father of microbiology. He was the first to observe, describe, and document living microorganisms, thus revealing the existence of an entirely new and hidden world of life forms, invisible to the naked eye. His pioneering work laid the foundation for microbiology as a scientific discipline. Leeuwenhoek made more than 400 microscopes in his lifetime. Each microscope had a single small lens fixed between brass plates. His lenses could magnify objects up to 275 times, which was much better than any other microscope of his time. Even though his microscopes were simple, they showed very clear and detailed images that no one had seen before. His skill in making powerful lenses became an important step in improving microscopes and helped scientists study tiny living things in greater detail. In 1676, Leeuwenhoek was the first person to observe and describe bacteria, which he referred to as animalcules. He discovered these microorganisms while examining material scraped from his own teeth, as well as samples from rainwater, pond water, and various infusions. The bacteria he described included forms that we now recognize as Koch, spherical, bacilli, rod-shaped, and spirilla, spiral. Leeuwenhoek also discovered protozoa, or single-celled eukaryotic organisms, when studying samples from pond water. He was the first to accurately describe various protists, such as paramecium and amoeba. His observations established the existence of an entire category of microscopic life, previously unknown to science, earning him recognition as the founder of protozoology. Leeuwenhoek made detailed observations of red blood cells, RBCs, noting their shape and size in human and animal blood. He was the first to describe sperm cells from humans and other animals, suggesting that fertilization involved the union of egg and sperm. He also studied muscle fibers, insect eyes, plant tissues, and mold filaments, contributing to early knowledge of cell structure and function. Leeuwenhoek had no formal scientific training, yet he followed a remarkably disciplined method of observation and documentation. He communicated his discoveries through over 300 letters to the Royal Society of London, accompanied by meticulous drawings and descriptions. His observations were verified and published in the Philosophical Transactions, gaining international attention and credibility. His detailed reporting established standards for scientific observation, replication, and communication that remain cornerstones of modern science. His work paved the way for later scientists, like Louis Pasteur and Robert Koch, to develop the germ theory of disease. He also influenced the development of cell theory and the evolution of optical microscopy. His meticulous curiosity and craftsmanship demonstrated that great scientific discoveries could come from simple observation and perseverance.
Louis Pasteur was a French scientist whose discoveries changed the world of medicine, food safety, and science. One of his earliest and most important findings was the discovery of molecular asymmetry. He studied the behavior of certain chemical compounds called tartrates and noticed that, although they had the same composition, they interacted differently with polarized light. He discovered that this was due to the asymmetric shapes of their crystals, some twisted to the right and others to the left. When both forms were mixed, they canceled each other's optical effects. This discovery revealed that molecules could exist in mirror image forms and laid the foundation of stereochemistry, proving that asymmetry is a key property of living matter. His studies on fermentation also changed scientific thinking. He found that fermentation was not just a chemical reaction, but a biological process carried out by living microorganisms. While studying butyric acid fermentation, he observed that the process stopped when air oxygen was introduced, a phenomenon now known as the Pasteur effect. From this, he introduced the terms aerobic organisms that need oxygen and anaerobic organisms that live without oxygen. This discovery helped explain many natural processes like decay and fermentation, showing that microorganisms play a major role in them. He also applied his knowledge of microbes to solve practical problems in the wine and beer industries, which were struggling due to spoilage. In 1863, at the request of Emperor Napoleon III, Pasteur investigated and found that unwanted microbes caused contamination. He discovered that heating wine or beer to 50-60 degrees Celsius could kill these harmful organisms without changing the quality of the drink. This method, known as pasteurization, revolutionized food preservation and is still widely used today to make milk and other foods safe to consume. In the mid-1900s, France's silkworm industry faced a crisis due to a mysterious disease that was destroying silkworm eggs. Despite knowing little about silkworms, Pasteur accepted the challenge. After years of careful research, he identified the microbes responsible for the disease and developed a method to select healthy eggs and prevent infection. His solution saved the silk industry in France and Europe, and his method is still used today in silk-producing countries. One of his biggest contributions was showing that tiny living things, called microorganisms or germs, are responsible for causing diseases and fermentation. Before his time, people believed that life could appear on its own from non-living matter, but he proved this wrong through his famous swan neck flask experiment. He showed that microbes come only from other microbes, which helped establish the germ theory of disease, the idea that specific germs cause specific diseases. Pasteur's greatest achievements came from his work on vaccines, which laid the foundation of immunology. In 1879, while studying chicken cholera, he accidentally discovered that old, weakened cultures of the bacteria lost their ability to cause disease but could protect chickens from future infections. This led him to the idea of using weakened, attenuated microbes to create immunity. He successfully applied this principle to develop a vaccine for anthrax in 1881. In a famous public experiment, he vaccinated sheep with his new vaccine, and when both vaccinated and unvaccinated sheep were exposed to anthrax, only the vaccinated ones survived, proving his theory. Pasteur's final and most famous success was the rabies vaccine. He believed rabies was caused by a microbe, although it was later discovered to be a virus. Since it could not be seen under a microscope, he developed a new technique using rabbits to pass the infection and then weakened the disease-causing agent by drying the spinal cords of infected animals. On July 6, 1885, Pasteur used this vaccine to save a 9-year-old boy named Joseph Meister, who had been bitten by a rabid dog. The boy recovered, and this marked the beginning of preventive medicine. The success of his rabies vaccine brought Pasteur worldwide fame and led to the founding of the Pasteur Institute in 1888, which became a center for medical research and vaccine development. Louis Pasteur's work not only saved countless lives but also laid the foundation for modern immunology and preventative medicine.
Robert Koch, a German doctor whose discoveries changed the way we understand and fight diseases. In the late 1800s, Robert Koch made several important contributions to bacteriology and pathology, the study of bacteria and diseases. He was among the first to show that specific microorganisms cause specific diseases. He developed new laboratory methods, including ways to grow and observe bacteria. Koch introduced the hanging drop technique, which allowed scientists to see living bacteria under a microscope. He also developed solid culture media using gelatin and later agar, which helped isolate and grow pure colonies of bacteria. Koch's experiments on wound infections showed that a unique microorganism causes each disease. He later worked with his team in the Imperial Health Office in Berlin, where he set up one of the first bacteriology labs in the world. There, he formulated his famous Koch's postulates, 4 rules that help prove that a particular microbe causes a particular disease. These postulates became the foundation of modern microbiology. One of his most significant accomplishments was identifying Mycobacterium tuberculosis in 1882, the bacterium that causes tuberculosis. With this discovery, he proved for the first time that tuberculosis is an infectious disease rather than a hereditary one. In 1876, he identified Bacillus anthracis as the bacterium responsible for anthrax, and in 1883, he discovered Vibrio cholerae as the cause of cholera. During his investigations in Egypt and India, he observed the comma-shaped bacterium and demonstrated that cholera spreads through contaminated water and food. These discoveries not only explained how these diseases are transmitted but also paved the way for effective prevention and treatment strategies. Although his attempt to find a tuberculosis cure with tuberculin was unsuccessful, it later became important as a diagnostic test. Koch continued his research on other diseases like leprosy, malaria, and bubonic plague, helping scientists understand how infections spread. Robert Koch's careful experiments, new methods, and sharp observations laid the foundation for modern microbiology and medical research.
Alexander Fleming, the man who changed the course of medicine with his discoveries. Fleming's scientific journey was marked by curiosity and careful observation. His first major discovery came in 1921, when he found an enzyme called lysozyme in body fluids like saliva and tears. One day, while suffering from a cold, a drop of his nasal mucus accidentally fell onto a culture plate of bacteria. When he checked it later, he noticed that the bacteria around it had been destroyed. This led him to realize that our bodies naturally produce substances that fight infection. Although lysozyme couldn't kill harmful bacteria, it helped scientists understand how the body protects itself from germs. But Fleming's most famous discovery happened by chance in 1928. After returning from vacation, he noticed that one of his petri dishes containing Staphylococcus bacteria had been contaminated by a mold. Surprisingly, the area around the mold was clear of bacteria. Fleming identified the mold as Penicillium notatum and realized it was releasing a substance that killed bacteria. He named it mold juice and then penicillin. This accidental discovery became one of the most important breakthroughs in medical science. Penicillin could kill many dangerous bacteria, including those causing infections like pneumonia and strep throat, without harming human cells. Although Fleming couldn't purify or stabilize penicillin himself, he understood its potential as a powerful medicine. Years later, during World War II, a team of scientists at Oxford University led by Howard Florey and Ernst Chain succeeded in producing penicillin on a large scale. It saved countless lives by preventing infections in wounded soldiers. In recognition of this work, Fleming, Florey, and Chain were jointly awarded the Nobel Prize in Medicine in 1945.
Sergei Winogradsky, often called the father of microbial ecology. He made several groundbreaking contributions that revealed how microorganisms play essential roles in maintaining life on Earth. One of Winogradsky's most remarkable discoveries was chemosynthesis. In 1887, he showed for the first time that certain bacteria can generate energy not from sunlight, but by oxidizing inorganic substances, a process now known as chemolithotropy. As part of his research on the sulfur cycle, Winogradsky identified sulfur-oxidizing bacteria such as Beggiatoa. He demonstrated that these colorless bacteria obtain energy by converting hydrogen sulfide into elemental sulfur and then into sulfuric acid, all in the absence of light. This discovery transformed our understanding of life by showing that organisms can survive and grow in environments without sunlight, such as deep-sea vents, by using chemical reactions instead of photosynthesis to obtain energy. Winogradsky also made major discoveries about the nitrogen cycle. He isolated and described bacteria such as Nitrosomonas and Nitrobacter, which convert ammonia into nitrite and then into nitrate. Through this, he proved that microorganisms drive these essential steps of the nitrogen cycle, which helps plants grow and supports all life on land. In 1893–95, he also discovered Clostridium pasteurianum, an anaerobic bacterium, i.e., able to grow in the absence of oxygen, that is able to utilize free nitrogen from the atmosphere in metabolic processes. He also invented something remarkable called the Winogradsky column, a simple glass column filled with mud and nutrients. This model showed how different types of bacteria organize themselves into layers depending on the availability of light, oxygen, and nutrients. It was a small ecosystem in a bottle and became a classic tool for studying microbial communities and ecology. The column is essentially a glass jar or cylinder filled with mud, water, and nutrients such as calcium sulfate and calcium carbonate. along with organic materials like newspaper or leaves. When left exposed to light for several weeks, the contents of the column naturally separate into distinct layers, each representing a unique microbial community adapted to specific environmental conditions. The top layer, rich in oxygen and light, supports photosynthetic organisms like cyanobacteria and algae, while the middle layers contain purple and green sulfur bacteria that perform photosynthesis without oxygen, using hydrogen sulfide as an energy source. The bottom, dark layer houses anaerobic bacteria that break down organic matter and release hydrogen sulfide, completing the sulfur cycle. This miniature ecosystem provided the first visual and experimental evidence of microbial stratification, showing how different bacteria occupy specific niches based on their energy and oxygen requirements. The Winogradsky column also illustrated key ecological principles such as biogeochemical cycling and energy flow in microbial communities. Even today, it serves as a valuable educational and research tool, helping scientists and students alike understand the complexity and interdependence of microbial life within ecosystems. Through his studies, Sergei Winogradsky revealed that microbes are not just disease-causing organisms, but also powerful engineers of the environment, recycling nutrients and sustaining ecosystems. Nicolas Appert, who is often called the father of canning, his work completely changed how humans preserve and store food. In the early 1800s, when food spoilage was a serious problem, Appert discovered that food could be preserved by sealing it in airtight glass jars and heating it. This process destroyed the microorganisms that caused food to spoil, allowing it to stay fresh for long periods without refrigeration. His method became known as appertization, after his name, and it laid the foundation for modern canning technology. In 1810, Appert published his famous book, The Art of Preserving Animal and Vegetable Substances, in which he explained his method so that others could benefit from it. His technique was first used to provide safe and nutritious food for Napoleon's army and navy, allowing soldiers and sailors to travel long distances without food going bad. Although Appert didn't know about bacteria or germ theory at the time, his process introduced the important concept of heat sterilization, which scientists like Louis Pasteur later explained. For his invention, the French government awarded him 12,000 francs, recognizing his enormous contribution to public health and food security. Nicolas Appert's discovery laid the foundation for the modern food preservation industry, and his work continues to benefit billions of people around the world even today. Joseph Lister, the man who revolutionized the field of surgery and laid the foundation for modern antiseptic techniques. In 1867, Joseph Lister introduced the antiseptic principle in surgery. He was the first to apply Louis Pasteur's germ theory to medical practice, realizing that microorganisms were the cause of infections after surgery. Before his time, surgical wounds often became infected, and many patients died, not from their injuries, but from infections afterward. To prevent this, Lister pioneered the use of carbolic acid, also known as phenol, as an antiseptic. He used it to sterilize surgical instruments, clean wounds, and spray it in the operating room. This chemical killed germs on contact and created a safe, sterile barrier between wounds and the surrounding environment. His antiseptic methods drastically reduced infection and death rates in hospitals, and his ideas quickly spread across the world, completely transforming surgical education and practice. Lister's work became the cornerstone of modern sterile surgical procedures that we still follow today. His legacy continues to live on. Even the well-known mouthwash Listerine, created in 1879, was named in his honor, reflecting his lifelong contribution to antiseptic science and hygiene. Edward Jenner, a brilliant English physician who is often called the father of immunology. In the 18th century, smallpox was one of the deadliest diseases in the world, killing millions and leaving survivors scarred for life. Jenner noticed something remarkable. Milkmaids who had once caught cowpox, a mild disease from cows, did not get smallpox. Curious about this observation, he decided to test it scientifically. In 1796, Jenner performed his famous experiment. He took material from a cowpox sore on a milkmaid's hand and inoculated it into an 8-year-old boy named James Phipps. Later, he exposed the boy to smallpox, and the boy did not develop the disease. This was the first ever successful vaccination, a term Jenner coined from vacca, the Latin word for cow. Jenner's discovery proved that exposure to a harmless form of a disease could protect against a deadly one. Because of Edward Jenner's courage, curiosity, and scientific vision, smallpox became the first disease ever to be completely eradicated from the world in 1980, a true testament to his lasting legacy. Paul Ehrlich, whose pioneering research laid the foundations of immunology, hematology, and chemotherapy. He was a German physician and scientist. From a young age, he was fascinated by colors and dyes, and this curiosity eventually became the key to some of his most important scientific discoveries. One of his earliest contributions was the development of special staining techniques to differentiate different types of blood cells and bacteria under the microscope. These methods revolutionized diagnostic medicine and are still used in modern laboratories today. Ehrlich's groundbreaking work in immunology began when he proposed the side chain theory, explaining how antibodies recognize and neutralize specific antigens. This theory became the cornerstone for understanding the immune response and paved the way for the development of vaccines and antibody-based therapies. He also worked closely with Emil von Behring on the diphtheria antitoxin, which was one of the first successful immunotherapies. Their collaboration saved countless lives and marked the beginning of a new era in medical science. However, Ehrlich's most famous achievement came with his search for what he called the magic bullet, a compound that could specifically target disease-causing organisms without harming the human body. This quest led to the discovery of salvarsan, compound 606, in 1910, the first effective treatment for syphilis. This discovery marked the birth of chemotherapy, using chemical agents to treat infectious diseases. For his monumental contributions, Paul Ehrlich received the Nobel Prize in Physiology or Medicine in 1908, shared with Elie Metchnikoff for their work on immunity. Selman A. Waksman was a Ukrainian-born American microbiologist whose groundbreaking research changed the course of medical history. In 1941, he coined the term antibiotic to describe small natural molecules produced by microorganisms that can inhibit the growth of other microbes. This simple word became one of the most important concepts in modern medicine, forming the foundation for how we fight bacterial infections today. One of Waksman's most remarkable achievements came in 1943 when he and his student Albert Schatz discovered streptomycin, the first effective antibiotic against tuberculosis. Until then, tuberculosis was one of the deadliest diseases in the world with no reliable cure. Streptomycin gave humanity hope and saved millions of lives, marking a turning point in the fight against infectious diseases. But Waksman's contributions didn't stop there. He and his research team went on to discover over 15 different antibiotics, including actinomycin, clavicin, streptofricin, gricin, and neomycin. Waksman's pioneering work also emphasized the importance of soil microorganisms, particularly those from the genus Streptomyces, as rich sources of antibiotics. His discoveries not only transformed medicine, but also laid the foundation for the entire pharmaceutical industry's antibiotic research. For his outstanding contributions to humanity, Selman Waksman received the Nobel Prize in Physiology or Medicine in 1952. His work continues to inspire scientists around the world to explore nature's potential for healing and discovery. Anand Mohan Chakravarthi, a pioneer in the field of genetic engineering and biotechnology, his groundbreaking work laid the foundation for modern microbial biotechnology and bioengineering. He pioneered plasmid transfer technology, showing how genetic material can be moved between bacteria to create new combinations of traits. This discovery became a cornerstone of modern genetic engineering, enabling scientists to design microbes with specific capabilities for medicine. Agriculture and environmental applications. Anand Mohan Chakrabarti and his colleagues in 1980 developed a genetically engineered bacterium, often called a superbug. This bacterium was designed to break down multiple components of crude oil, including camphor, octane, xylene, and naphthalene, which are major pollutants in oil spills. Chakravarthi used strains of the bacterium Pseudomonas, each naturally capable of degrading one type of hydrocarbon, like xylene or naphthalene. Using plasmid transfer technology, he combined the plasmids, small circular DNA molecules, from these different strains into a single Pseudomonas strain. This new bacterium inherited the ability to degrade multiple hydrocarbons simultaneously, making it much more efficient at cleaning up oil pollution. The bacterium was called Pseudomonas putida, often referred to as the oil-eating superbug. Chakravarti's work also made history in the world of law and innovation. His oil-eating bacterium led to the landmark 1980 U.S. Supreme Court case, Diamond v. Chakravarthi, which ruled that genetically modified organisms could be patented. This was the first patent ever granted on a living organism and established the legal foundation for today's biotechnology industry, affirming that anything under the sun made by man could be protected by patent law.